Good morning to everybody. I'm introducing myself. My name is Fabio Apone. I'm the scientific director of Arterra Bioscience and Vita Lab, which are two Italian companies involved in the development and commercialization of cosmetic active ingredients and other type of ingredients having some dermatological applications. So this is the title of the talk today. I want to show you some of the most recent results we obtained on one active ingredients and the validation of its cosmetic efficacy by using a dermal equivalent model. So as you know, UV radiation are of course very dangerous for the skin, and under the name of photoaging, photoaging includes a complex, set of phenomena. They are linked to chronic exposure to UV light, and causes a lot of serious damages and dramatic morphological changes to the skin. Besides, inflammation or decrease of collagen, the content of collagen, increasing collagen fragmentation, we have also a lot of damages at the level of the cells that compose the skin, both fibroblasts and keratinocytes, which alter the metabolisms and functionality. That's why a lot of companies, and in particular also cosmetic companies, are investing a lot in searching for new products having these characteristics of protecting the skin from UV light, because it's a very relevant issue for cosmetic and dermatology. So. But these active ingredients, of course, have to be safe and effective in order to be, launched on the market. They have to be guaranteed for two of these most important characteristics. Vita Lab is working a lot on plant natural extracts. It's a biosustainable company. It means that it's produces green type of active ingredients. And cell cultures are an important source of active ingredients. The plant can be easily manipulated in laboratory nowadays. I'm not talking about genetic manipulation, but in particular they can be in terms of tissue culturing. I mean, that means each part of the plant can be cultivated on a solid media, like here in the picture, and a mass of cells can be produced from this part of the plants, each part of the plant, producing like calluses, like you can see in the picture below, which are masses of undifferentiated cells that have totipotency, have several potentialities. These cell cultures, these calluses can be then put to grow in liquid, as liquid suspension cultures in flasks or bigger containers. So the process can be upscaled in order to obtain big mass of cells that can be used to extract products, compounds, or making extract. Why using this system? Why using plant cell cultures? I listed here some of the advantages. There are several advantages using plant cell culture compared to plants grown in the field, for example. First of all, we always have a continuous supply of material and in standardized conditions. That means that always there's a high level of consistency from batch to batch. The materials that we obtain is always safe and clean, means that there is no risk of environmental contamination or risk of pathogens present in the extracts. It's very sustainable, and that's actually one of the most important concerns also for cosmetic companies. Biosustainability. It means that there's no exploitation of agricultural land. The system is versatile. The compounds can be increased by using elicitors or by using genetic or biochemical tools. In many cases, the compounds are produced in higher amounts than in the plants, and the extraction process is usually much easier than the plants. So that's why the cosmetic company, including Vita Lab, are investing in using this kind of system, these cell cultures, to extract cosmetic active ingredients. And also because the plant cell culture can be used as biofactories, means that they can use to produce a certain type of secondary metabolites. They're usually present in the different organs of the plants. The example I want to give you in the talk is the one of Dolichos biflorus, a cell culture extract, which it was recently developed by our company. Dolichos biflorus is a plant, is a bean plant that is particular grown in tropical, subtropical area of the world because it's one of the main dietary protein source. And the plants contain several compounds with proven activity, therapeutic and beneficial effects on human cells. So the extract we produced, the plants were used to develop a cell culture system, and from cell culture we produced this hydrosoluble extracts. And the chemical analysis of the extract produced from the cell culture of Dolichos biflorus revealed the presence of several compounds. This is one of the antioxidant profile. Many compounds are like genistein, diethane, and other glucosidic derivatives that already in literature are known to have therapeutic properties in protecting the cells from senescence and from UV radiation. So they have, due to their strong antioxidant activity, they are good in the photoprotection. So based on this assumption, what we did, we used this extract of Dolichos cells to test on the cells, on cultured cells as first. And we used cultured keratinocytes and the fibroblasts, primary keratinocytes and primary fibroblasts from human skin. And for keratinocytes, here the first graph here, we observed I can use the point actually, it's much easier. For cultured keratinocytes were induced to produce cytokinins that are the inflammatory molecules, compounds for these cells. Interleukin 1 beta, 6 and 8 were increased by the UVB radiation. And the effect of the extra was to significantly reduce, to kind of abolish the effect of induction of the cytokinins production. Similar effect Dolichos extra had on fibroblasts, in this case were irradiated with UVA light to induce the production of metalloproteinases. Here reported metalloproteinases 1 and 3 as the most important effect. And also the extract had the effect of reduction of the expression of these proteins. Similarly to this positive control that we use here in the keratinocytes was a drug known for its anti-inflammatory properties. For the metalloproteinases, we use retinoic acids also because it has an effect of reduction of metalloproteinase expression. At the cellular level also, we observed that Dolichos extra had the significant effect of reduction of the damage at the level of DNA. Here shown we measure the level of fragmentation at the nuclear to the nuclear DNA done by the COMET assay, which is a common assay used for this type of analysis. So UVB induced the fragmentation of nuclear DNA that was reduced by the extract, similarly to the Trolox, which was an analog of vitamin E used as a positive control. And the same thing happened for the mitochondrial DNA damage, where we use this common deletion assay to also measure the damage level of the mitochondrial chromosome. Also in this case, Dolichos extra had those dependency effect of reduction of the damage to mitochondrial DNA. So the in vivo test confirmed the results we obtained on cell cultures. That means that Dolichos had both a preventing effects of erythema formation, also a soothing, a curative effect. In this case, the volunteers were first treated with the extract for 1 week, and then were irradiated with a mixture of UVA with UV light. The erythema formation was induced, and in the volunteers, in the area of the skin of the volunteers who were treated with the extract, we observed a reduction after 24 hours of erythema formation that confirmed the preventive effect of photoprotection of Dolichos. And the similar results we obtained for the curative, we say, soothing effect. In this case, the volunteers were irradiated with UVA and UVB light to induce erythema formation. After 24 hours here, there was a treatment with the extract. After 2 hours and 4 hours, we observed the reduction of redness. And this reduction was significant of around 13% compared to control, the untreated control, here around 24% compared to the control and the placebo. So, of course, the cell system and the in vivo tests are very useful to measure, to analyze the effect of active ingredients on the skin, on the skin cells in terms of gene expression for cell culture, in terms of final effect or in vivo test. But to measure, if you want to measure and visualize the real effect of damage at the level of collagen architecture, for example, organization, we need to use another alternative system. That's why we developed another system, a skin equivalent model. Other companies use already artificial skin model or ex vivo explants to do this type of analysis. What they usually do is to put fibroblasts in presence of a collagen, then they wait 3 days for the contraction, and then they add the keratinocytes on the top. In our system, it's different because that was realized in collaboration with the University of Naples, the Department of Bioengineering. We use some microcarriers, gelatin beads, that are put in the presence of fibroblasts. So fibroblasts are seeded on these microbeads in order to, and then induced, to proliferate, to produce all the proteins of the extracellular matrix of the skin, including collagen, in order to obtain a tridimensional structure like that. And then on the top of this dermal equivalent, we can add the primary keratinocytes in order to have a complete system. For this study involving the Dolichos extract, we actually use only the dermal equivalent model, the one that was produced only by the fibroblasts. Here is shown in the picture. Here some section with staining, where still are visible some of the gelatin microbeads after 2 weeks. And then they slowly disappear. They get degraded after 4 weeks. Because we want to actually analyze the effect of UV and of Dolichos protection at level of the dermis, okay? Dermis and collagen architecture. So I'll show you some of the results. In this dermal equivalent model we call DIM, we can do this type of analysis. What we did was to stain this section with picrocerius red, which is a dye that specifically stains collagen. But we can discriminate between 2 types of collagen fibers, because by using polarized light, we can obtain a green signal that corresponds to thin fibers of 0.8 or less. And the thick fibers give a red-orange signal. So on the basis of different use of colors, we can have different type of signals, different colors that correspond to different thickness of the collagen. And we can also analyze the damage level of this type of fibers. If we expose this dermal equivalent model to UVA light, for example, here after 6 hours of the treatment with the 20 joule per square centimeters, we observe a reduction both type of fibers. Thick collagen fibers get reduced, are degraded significantly by the radiation. It is are reported at the times of 6 hours and 3 days. Okay, so the thick collagen fiber, and the same thing happens for the thin collagen fiber. So we observe a reduction of the signal of the both the red and the green signal on the dermal equivalent model. So when we observe the protection that was performed and produced by the Dolichos extract, this DIM model were treated here with Dolichos extract at this concentration, and with retinoic acid, which works as positive control also for, it's a good protector, a photoprotector. After 6 hours and 3 days, we observed a significant, reduction of the thin collagen fibers that were abolished, totally abolished by the presence of the Dolichos extract and also the positive control retinoic acids. This effect was actually significant only on the thin collagen fibers, maybe because those are more sensitive to this kind of a treatment. And this effect was mostly due to the reduction of metalloproteinases. We measure, like similarly to what we did in our cell culture model. Also on the skin, on the dermal equivalent model, we measure the synthesis, so in this case the secretion, so the synthesis of metalloproteinases. Here it's shown in the picture for metalloproteinases 1, that we use the antibody labeled with cytone green, and we observed that the induction of metalloproteinase expression induced by UV light was decreased significantly by the presence of the extract. And here it's shown also in the picture. Metalloproteinases 1 at 3 days is visible, was significant effect. For metalloproteinases 3, both 6 hours and 3 days. So another interesting thing for us, assay and type of analysis that we performed was to measure the effect of coarsening on collagen. That means that after UVA radiation, in this dermal equivalent model, we can observe a type of effect of coarsening of collagen. Means that the fibers get kind of separated, get more loosey. And we can measure this type of effect by using this kind of this multi-photon microscopy. Well, we can obtain like correlation curves here at 6 hours. That means that the higher the curve, the more we can say coarse is the collagen. As you can see, the effect of UVA that increased this, the tendency of this curve is totally reduced, abolished by the treatment with Dolichos. Here, just reported the values in the graph, where at 6 hours and 3 days, this measurement revealed that the effect of coarsening of collagen induced by UVA light was totally reduced and abolished the presence of this treatment. And it's important to notice that this type of analysis that we perform, it's possible to do only on certain type of dermal model or skin model. Why? Because when we try to do this kind of analysis on another type of dermal equivalent model, for example, the one that was produced by the use of exogenous collagen and the fibroblasts that are then mixed, as you can see, the collagen fibers are all random and are not organized like in the dermal equivalent model that we used, that have a pattern of collagen bundles much more similar to the human skin sample, as you can see. So we can perform this type of analysis only on this type of dermal equivalent model and not on the other type. So we have of course some future development and perspective, and now we already realized the complete skin equivalent model by seeding the primary keratinocytes on the top of the dermal system in order to obtain a complete, skin equivalent system that's much similar to, it's resembled to the human skin samples for its kind of organization of the fibers and with the presence of basal lamina and so on. So this kind of system can be easily used also to assess the activity of new active ingredients having cosmetic and dermatological interests, which is mainly our focus. And we can use it also to analyze the response, for example, both dermis, epidermis to new active ingredients and their interaction, or to use to do some kind of studies that it's not possible to perform on other type of system like culture cells or in vivo systems, which are like permeation studies, hydration measure, or analysis of elasticity, mechanical stress that can be, facilitated and increased by the presence, by treatment of active ingredients. Just conclusion, this research was conducted by Vital Arterra Bioscience, which are the companies, a biotech company located in Naples, in collaboration with the Center of Biomaterials and for Healthcare located in Naples, Department of Bioengineering. This is the person involved in this study, in the project. And so thank you for your attention. And if you have any question, I'm happy to answer. Any type, each type of plant, so even if it could seem kind of very easy process to obtain plant cell culture, on the opposite, sometimes it's kind of hard and difficult because some plants are not very easy to be induced in the proliferation of these calluses. The media usually for the callus is solid media composed by agar and some nutrients. Of course, there are different types of media, several types of media, and we have to play with, for example, the presence of certain microelements or the hormones, phytohormones, in order to induce, of course. So you have to play with that, and it's not always the same for all the species.
So do you think it's a good idea to infuse the plant extract with the agar and ask the cells to grow there?
To use the plant, the media to.
Right.
What do you mean? To use the media as active
No,
ingredient?
just extract the plant components. I mean,
from the cells, yeah.
No, from the tissue. Take the tissue, extract it, and incorporate to the agar media, and then whatever the desired cells you want to grow or desired parts you want to grow, just put them on there and grow, because you are already providing whatever the micronutrients which is there in the plant.
No, actually, the procedures that you have, you need to have a sterile media with particular balance composition of elements. So usually are not made by extract from other plants. So the media has to contain sucrose, contain other micro elements that have to stimulate the growth. But no, usually it's not that the way to grow the plants. I mean, the cells need to have a special balance of all the elements and ingredients. That's the way all the plant biologists do. So.
Okay. So any part of the like root, fruit, or leaf, anything you can grow like that.
Anything can be used like a starting material to produce the undifferentiated cells. Yes.
Undifferentiated cells.
Yes.
So can we take the undifferentiated cells and subculture them?
Yes. Yes. We
so
can.
So ultimately,
you're talking about differentiated cells or undifferentiated?
Say, for example, you put the fruit or a root, and then differentiated cells are growing.
Yes.
Like in colonies.
Yes.
Right? So take one colony and subculture them.
Yes.
You can.
The way we do.
So ultimately, can you produce a cell line for that?
You produce a cell line, yes. That's why the process is very standardized. So it's a clone. So you produce a line.
It's a clone.
It's a clone that has all the same characteristics. So you can subclone until they get older. So you have to start again from the callus. But usually it takes a long time.
And with your Dolichos extract, you showed some gene expression, mRNA level, by like 60-fold, 70-fold increase or decrease. So do you have any protein data for that, ELISA, that IL-6, the pro-inflammatory cytokines especially?
The ELISA for the cytokines,
yeah,
for example?
To confirm the protein.
Yes. I didn't present the data. Yes, we have the data also, the cytokine production, expression in the media, because are secreted by the keratinocytes, and we can also measure the amount of protein there. Yes.
Nice. And with following to that, the extract, Dolichos extract you used, is there any active ingredient identified in that, or it's just a total extract of the undifferentiated cells?
It's a total hydrosoluble extract. So we take only the water-soluble part of the cells. So we, of course, have to get rid of the fibers, all the membranes, and the other components. We can have from the cells different type of extract. If we use different solvents, we can have different type. In this case, it's an hydrosoluble extra, total extra. So it contains amino acids, proteins, vitamins, everything. Yeah. Yes, plus the antioxidant compounds I showed in the graph.
So you showed some patients' data also. I mean, the volunteer data.
The volunteers, yes, we do this.
So and you
we have this test done actually by another company that make in vivo tests.
I see.
We don't do by ourselves.
So, with that, did you by any chance analyzed for the long-term effect of your extract?
No, it was analyzed only on the erythema formation, so the times the one I showed. For the preventive effect, it was a treatment from 1 week to 10 days. So after 1 week of treatment, then there was irradiation, and then it was measured the erythema formation for 24 hours and the reduction over time. Okay, so that's the longest time we have this data. I mean, we didn't do prolonged, because erythema actually appears after 24 hours. So it's.
Okay, I might be completely wrong. I think I mean, animal studies will be a good idea to see what is the long-term effect of this.
Long-term. What do you mean more long-term? How long?
Long-term means if you are using for years together in the same patient.
Okay, no, okay.
So that is the long-term
Maybe
effect. That you can, I think, easily confirm by the animal studies, where you can apply
throughout
the life of
you mean
the
over
animals.
Years? It's kind of study we may think to do. Why not? Because it would be interesting to look at it, but we haven't done yet.
Thank you. With this.